So I thought that I would start with a little bit about my background. So I got my bachelor's in maybe a completely different field from what I'm doing now, in fisheries and marine sciences. I majored in oceanography and marine geology specifically. I did my bachelor's in Sri Lanka, in University of Ruhuna, in this beautiful campus right next to the sea. Then I moved to China, to University of Chinese Academy of Sciences, to do my master's. So this was a 3-year full-time research program where I spent a lot of that time in South China Sea Institute of Oceanology in Guangzhou, conducting research on deep sea fungal natural products. Then I moved to UK to do my PhD at the University of East Anglia, where I'm now a 3rd-year PhD student at the Genomic Centre. And my research here focuses on employing bioinformatics approaches to find new antibiotics from soil Pseudomonas. So before I start, I wanted to explain why we need new antibiotics. To do that, I want you to have a look at this image, where we see when each antibiotic class was first discovered and when we first saw resistant strains emerge. So it started with the penicillins in around 1928, which is the first antibiotic isolated from natural sources. And we see the resistance emerging just after 12 years. And for each antibiotic along that timeline, we see this trend has persisted, and we've seen the resistance emerging just a few years after, and that window is getting smaller and smaller with time. And with that, AMR, or antimicrobial resistance, has become one of the biggest problems we have to address in recent times. And to worsen the situation, for nearly 3 decades now, we have failed to discover new classes of antibiotics. But the pathogens continue to develop resistance to existing antibiotics. There are several reasons for this gap. First being that developing new antibiotics is very costly. This is also worsened by the fact that antibiotics are given for a short duration of time. They're not long-term drugs. So the companies who develop antibiotics do not make a lot of profit. The other problem is that there's strict regulatory standards and therefore long development timelines, and one antibiotic takes about a decade to come to the market. Because of these 2 reasons, the companies, especially the private pharmaceutical companies, do not want to invest in antibiotic development because there's a low return on investment. And this is kind of the financial side of the problem, which is not to be ignored, but we do have even more important issue than that, which is maybe as researchers what we can address on our part are the scientific challenges in discovering new antibiotics. So when it comes to antibiotics, more than half of the drugs that we use today come from natural sources, and majority of them have bacterial origins. Therefore, bacteria is kind of a good place to look for these new molecules. But the issue is that they don't always make these products, since they are secondary metabolism and not required for general maintenance of the cell. So you only see low levels of production under standard laboratory conditions. And for certain compounds which can only be produced under specific conditions, you might not see the production at all. So that makes it difficult to detect with traditional screening. And the second problem would be high rate of rediscovery. If you remember the timeline I showed you before, there from 1940s to 1960 was considered as the golden era of antibiotics. That's when most of the antibiotic classes that we use today have been covered and introduced. This is when the most of the low-hanging fruits were picked, and now we are kind of left with the more harder to find antimicrobials. So what happens under conventional methods is that we rediscover the same molecules. And the third challenge would be the lack of cross-disciplinary collaboration. What we'd like to see is that to address those problems, we need different disciplines coming together, such as organic chemistry, analytical chemistry, genomics, bioinformatics coming together to solve such problems, but we don't see enough of that happening. So this is where my project comes in, trying to answer this fourth problem by developing an antibiotic discovery pipeline, which goes from environmental isolates to sequence analysis with various bioinformatics tools that we have access to. And then to study the secondary metabolism of bacteria, then validating the predictions of that pipeline, all the while reducing the chance for rediscovery. So my project starts with isolates from agricultural soil and high throughput screening for bioactivities. So our lab has a large collection of environmental isolates of Pseudomonas from the studies of different people who's worked over the years in the lab. So I used a fraction of that for my project. I started with 1,248 strains and screen them over human pathogens like E. coli, Pseudomonas aeruginosa, and MRSA. And based on those activities that we saw, we selected 93 candidates to go forward with. These isolates had varying ability to inhibit pathogenic strains, ranging from broad spectrum activity to a set of strains that didn't have any activity at all. So these 93 strains were then sequenced. We did both long and short read sequencing to get hybrid assemblies. And then these were subjected to the genome mining pipeline. I walk you through this pipeline step by step. Some of you may be familiar with some of these tools. So we start with the assembled genomes, and then annotated those genomes with Proca. For genome mining, I primarily used Anti-Smash. There are other tools that you can use like NP Searcher or Bagel or Ripper, but Anti-Smash is like a all-in-one kind of a tool. So and it's kind of more popular as well for bacterial genomes. I think they do have both fungal and plant versions of it, but what it basically does is scans for conserved domains in the annotated genes by comparing them to the domain models of in databases such as PFAM or TigerFAM, and Anti-Smash also has its own domain library to refer. So these biosynthetic domains are often specific to certain biosynthetic pathways, and that allows it to recognize the type of secondary metabolite it's produced. As for the boundaries of these gene clusters, Anti-Smash makes kind of a learned guess based on the fact that a cluster should contain core, tailoring, and transport-related genes. And these are typically co-located and co-expressed. So once this is done, if I show you an example with 3 strains, we have different regions of the secondary metabolic gene clusters along the genome. And what you have as the output is a separate GenBank file for each of these regions. So you can then submit the whole output of Anti-Smash to Bigscape. So what Bigscape does is that it begins by parsing the biosynthetic domains within each biosynthetic gene cluster and compares individual domains across the BGCs to quantify the similarity between gene clusters based on the shared biosynthetic machinery. It then calculates a similarity scope based on the domain content, gene order, and functional similarity. And then using that construct similarity networks. So for this specific example, it would look something like this, because we have 4 A-type clusters. So it has put that into a separate gene cluster family. We have 3 B-type clusters, so it has put that into a separate gene cluster family. So this is kind of the example. But for my data set, since I have 93 genomes, my sequence similarity network looks something like this. So there's 929 gene clusters here and 162 distinct gene cluster families in this data set. I've marked all of the known clusters here. One thing that I forgot to mention was that in the pipeline, you might have seen the MI-big annotations coming in. When you run anti-smash, anti-smash will also give you, if any of your clusters are similar to an MI-big entry, it will tell you that it is and how much similar it is. Also, when you run a Bigscape, you can also add the MI-big flag, so that when it make those networks, it will pop up like the MI-big entries if there's anything similar. So here you see that the majority of these gene cluster families do not have known gene cluster matched to it. So most of them are novel gene clusters. And also the colors here are specific to certain biosynthetic pathways. And you see that here on the left side, that NRPS clusters, that those are the non-ribosomal peptide synthesis, they have the most diversity than the other pathways. This is kind of very typical of Pseudomonas secondary metabolism. If you see the past studies of this nature, you see similar networks coming up for Pseudomonas. Yeah, so the next is that if you map that similarity network into a phylogenetic tree, this is what it would look like. And here on the left side, you have the phylogenetic tree, and each filled circle here is a presence of that certain gene cluster, and you can see how they are distributed. And at the end, on the right side, are the pathogenic inhibitions and all of the presences of inhibitions here. And the colors here are also consistent with the previous similarity network. Essentially, what I wanted to point out here is that this is a data set with presence and absence for all of the variables being the gene cluster families or the inhibitions. So these are pluses and ones and zeros. Another tool that I want to talk about is cBlaster, which I've used in that pipeline. This is a tool for finding clusters of co-located homologous sequences. It is a very useful tool to find gene cluster regions that are known but not detected by anti-smash. You can give it a collection of protein sequences, and cBlaster can search sequence databases remotely through, if you want it to do remotely, it can search through via NCBI Blast, or locally, which is what I've done, if you have a database with your own sequences. And this is very useful for finding the known clusters, if any of your strains have them, because anti-smash is not perfect. It sometimes cannot identify certain gene clusters. Like, for example, until very recently, it couldn't detect the hydrogen cyanide cluster or tropolone cluster. But it's improving. But then again, you have this kind of tools that you could use for in instances like that. So I've used this to supplement my data set as well. And we come to the next and the final step of my genome mining pipeline. It is to correlate those presence and absence of the gene cluster families with the activity. And here is the correlation heat map of where you see positive correlations in pink. To simplify things, we are only going to focus on this region here. where you see the correlations between gene cluster families with my pathogen inhibitions. So also that region, I'll show you the top 10 predicted or correlated gene clusters for each of the pathogen inhibitions. And in the interest of time, I'm going to show like 2 pathogens. And one thing that I want you to focus are the ones that are highlighted here, because they are the ones that are known, that we've confirmed that these are actually present in those strains. So for PA01, Pseudomonas aeruginosa PA01 strain, we have top 10 gene clusters here. And you see the highlighted ones. If I go through one by one, here promycilline is a broad spectrum antibiotic. Its mode of action is cell wall disruption. And pseudopyranine is also a potent antimicrobial. Butysolvine, it's a lipopeptide. It has the ability to inhibit and break down biofilms. And carotenoid does also reported to have antibacterial properties. And here histiocorgatine, piverdine, picaline, these are all siderophores. These are ion chelating molecules. They can give an antibacterial effect by limiting iron for nearby organisms. And nunomycin is also another antimicrobial cyclic lipopeptide. And for MRSA, we have these clusters coming up as the highly correlating ones, and you recognize some of the products that we've talked before, and also there's a hydrogen cyanide cluster coming up as well, which is probably self-explanatory. One thing that I want to point out here is that a lot of already known antibiotic producing gene clusters or compounds that could give you a biocontrol has come up in the correlation and as targets in this pipeline, which is very encouraging, as that means that the strains indeed contain this genetic material to explain their bioactivities, and this kind of pipelines can accurately captures them. And we can be hopeful that any unknown clusters that is predicted may also be responsible for these activities we see, and is worth looking at. So this approach kind of gives us a roadmap. And if you remember, I had like 900 gene clusters before, and this kind of approach actually reduced that amount to a handful of gene clusters for us to study and validate. And all the while, this is also useful because we know which known gene clusters are present, and we can then overlook those known gene clusters and then go for the novel looking gene clusters that would actually help us reducing the rate of rediscovery. So the next step of my project, which is where I'm currently at, is the experimental validation of the target predictions that I got from this pipeline. These are some of the genomic and analytical methods that I use to validate. First being the gene knockouts, where we knock out a critical gene in the biosynthetic pathway to confirm the relevance of that certain cluster to the activities we see. Another way to do this would be transposon screening. This is more useful when you have a strain which has multiple predicted gene clusters, and you can use a transposon library to screen on those multiple conditions. I'm also using analytical methods for screening as well for strains where you see biocontrol but cannot really pinpoint which cluster might be responsible, especially in the case of rare clusters or singletons, which the clusters that present in only one strain, where the correlation strength might be too low, and there's a chance that we might, overlook those clusters. So this way you can detect the molecules first and work your way back to the gene cluster. And hopefully, on the way, we'll find some new antibiotics. So this is kind of where I would like to conclude. And with that, Thank you very much for listening to me. I should also thank all of my lab members, my supervisors and funding, also to GetGenome. Especially thank you, James, for giving me the opportunity to talk today. And I'll be happy to take any questions if you have. Thank you.